Okay, so let's start working on reproductive system. Chapter 27, this is part A. If I remember, we do male repro before female in here. Okay, understanding the anatomy and physiology of the reproductive system will give you the ability to recognize symptoms like sexually transmitted infections, give you the ability to treat patients with reproductive diseases, help you educate patients on how to have a healthy reproductive system. As always, let's jump into the little video. Okay, so let's go in and start talking about the anatomy here. The reproductive system does not function continuously because we're not constantly reproducing. That's pretty straightforward. It doesn't become active until puberty. Male and female reproductive organs are different, but share 4 tasks. I don't know why they broke these out into 4, but let's read them. Okay, so 1, form gametes. Well, okay. Reproductive cells, haploid reproductive cells. Bring gametes together through sexual intercourse. Combine genetic information contained within gametes through fertilization. And support the development of the fetus, gestation, and the birth of the baby, parturition. Okay, so gametes are haploid. That means they have half the normal amount of DNA. In humans, the typical number of expected number of chromosomes is 46. That's the normal number. But there are 23 chromosomes in a sperm, 23 in an egg, 23 plus 23 when they come together. That gives you 46, the normal number. So understand that these are not just making cells. It's not just making cells that are involved in reproduction. It's doing a specialized form of cell division to end up with these special haploid cells that we call gametes that have half the normal amount of DNA. All right, so we bring them together, bring the gametes together through intercourse. That's cool. Says combine the genetic information. When an egg and a sperm fuse, we call this a zygote. The zygote is the first cell of the new individual. Actually, we form a zygote, and then we're going to divide a few times until we have a morula, which up to about 8 cells, we're calling a morula, 16 cells maybe. Then we're going to do a gastrula. That's when it becomes a hollow ball of cells, and then we'll have an embryo, and then we'll have a fetus, and then we'll have a baby. Anyway, the support the development of the fetus, gestation, and the birth of the baby, parturition. Pretty straightforward. All right, so male and female structures are homologous. They're similar, they're derived from the same thing, but they differentiate during development. So yeah, where I say they're derived from the same thing, they have a common origin in development. So they're male testes, female ovaries, those are both gonads. And they come from the same tissue as an embryo, but which direction they go depends on how you differentiate. Male penis, female clitoris, same thing, similar structure, but similar structure derived from similar tissues, but not the same. They differentiate during development. All right. So the primary sex organs are called gonads. In males, we call those gonads testes, and females, we call those gonads ovaries. Gametes, that's the sperm made by the male, the egg made by the female. Gametes are formed through a special form of cell division we call meiosis. Sex hormones, steroid hormones, testosterones in males, and actually, that's not true. I don't like the way it's written at all. Testosterone is an example of a sex hormone that is more common in men. The male sex hormones are called androgens, and the female ones are called estrogens. Understand that testosterone is just one of the different types of androgens. Anyway, but yeah, estrogens and progesterone in females. The vital development and function of reproductive organs, other organ tissue, sexual behavior, sexual drives. Oh, yeah. You don't differentiate properly unless you have the correct hormones working. And I'll add to this because they're not mentioning it here. And you have the correct receptors for the hormones on the surface of the target cells. If you don't, you're going to develop differently, as though the hormone wasn't present in sufficient numbers, even if it is. All right, accessory organs. These are ones that are not the testes. Sorry, not the gonads. So we're talking about the ducts, the glands, the genitalia that we use for moving the gonads, or sorry, moving the gametes around. All right, so the hypothalamic pituitary gonadal axis, the production of gametes and sex hormones is regulated by a sequence of hormonal events involving the hypothalamus, anterior pituitary, and testes, assuming you have testes. Referred to as the hypothalamic pituitary gonadal axis. And the hormones that are involved: gonadotropin releasing hormone, follicle stimulating hormone, luteinizing hormone, testosterone, and inhibin. All right. So, going through that list of hormones, gonadotropin releasing hormone comes from the hypothalamus, and this is it goes to the anterior pituitary cells through the hypophyseal portal system. The hypophysis is neurohypophysis, adenohypophysis. We're talking about parts of the pituitary. We're still way up in the brain here. Follicle stimulating hormone, luteinizing hormone. These are gonadotropins from the anterior pituitary. All right, that's straightforward enough. The sex hormones, in males, we would use testosterone and estrogen. In females, we would use progesterone and in both cases, the new, exert negative feedback on hypothalamus. All right. So if we have enough testosterone, or if we have enough progesterone, we are going to send a signal to the hypothalamus and the anterior pituitary saying, stop that, we have enough. That's what a negative feedback loop does. Inhibin is released from the gonads of both males and females, and it has a negative feedback on follicle stimulating hormone in the anterior pituitary. Okay. Yeah. All right. So hold on just a second. Okay, so you guys can see this is pretty straightforward. The hypothalamus, which is up here, the hypothalamus makes the gonadotropin releasing hormone, which travels down through the infundibulum here, through the stalk, to the anterior pituitary. And there it tells the pituitary to make luteinizing hormone, follicle stimulating hormone. Those travel through the blood down to the gonads, either the testes or the ovaries. And the sex hormones that are present, along with inhibin, are going to affect the target cells that they divide and become. So, and let's see, where's my mouse? There's my mouse. All right. So the axis continued. Testosterone in males, estrogen, progesterone in females. These act on the target cells in the body, and those cells will exert negative feedback on the hypothalamus and the pituitary. Let's see. Inhibin is released in the gonads of males and females, and it is, just like we said a moment ago, exerting negative feedback on follicle stimulating hormone when released from the anterior pituitary. Okay. Yeah, so we've got that activation of the axis. Puberty is the stage of life when we become reproductively active. Those organs grow to their adult size and they become functional. We can't really reproduce before the organs can do the job. So now, before puberty, there are low levels of circulating steroidal sex hormones that can suppress gonadotropin releasing hormone. When puberty approaches, the hypothalamus gets less sensitive to the sex hormone inhibition, and because it's not as sensitive anymore, it doesn't respond as though there was a level of sex hormone to stop it. The hypothalamus will then release gonadotropin releasing hormone, and doesn't do it steady, does it in spurts. That'll cause the anterior pituitary to release follicle stimulating hormone and luteinizing hormone in response to when we have high levels of gonadotropin releasing hormone. Obviously, you probably guessed, if follicle stimulating hormone, luteinizing hormone are released whenever gonadotropin releasing hormone levels rise, they must be, drumroll, the gonadotropins. All right. So follicle stimulating hormone and luteinizing hormone will cause the gonads to release more sex hormones from the gonads, and the threshold for inhibition of the gonadotropin releasing hormone will go up higher. That means that you have to get higher, reach a higher level to cut it off. and that means that we don't reach it as easily. We don't cut it off, and more sex hormones are getting released. The sex hormones are released until an adult pattern of hormone interaction is achieved. Great. So meiosis is cell division that creates gametes. You know that mitosis is normal cell division. Mitosis is how we make the cells to repair wounds or to grow or to replace old worn out cells, things like that. Mitosis is what I just described, and it has you start with the normal amount of cells, normal amount of chromosomes in a cell. 46, you're going to double the DNA, and then you're going to divide in half. So you're going to end up doing one division and getting the normal amount of chromosomes. So you go from normal to doubled, and then you divide into two that are normal. Meiosis, which we're about to talk about, is only for making gametes, only for making eggs and sperm. In it, you start out the same. You have the normal amount of chromosomes. You double the amount. But the way you divide is different. You divide once, and that gives you back to the normal amount. But those then divide again, and you end up with 4 daughter cells, each of which is haploid, has half the normal amount of DNA. And as this PowerPoint says, there is a reason you wouldn't want to have 46 plus 46, and then you end up with too many chromosomes. So the reason we're doing this special division is to divide in half, lose half the DNA. All right, so non-sister chromatids. When we have homologous pairs, this would really be helpful if we had a picture to go with. Yeah, we do. Okay. So most of the hormones in the body, when we go through cell divisions, we're going to line up as two copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, etc. Sex hormones are not necessarily that way. Say you have an X and a Y because you're male. Those are not homologous. Okay. If we have 2 X's, then they are. Anyway, let's back up. So sister chromatids are yeah, back up. Why are we? Yeah, we're just going through basic terminology. So the sister chromatids are halves of a doubled chromosome. We start with a normal chromosome. We make a copy of it that's still attached to it. So normal chromosome, make a copy that stays attached. We now have a doubled chromosome. What we were calling a chromosome, we now call a chromatid. And so this over here is a chromatid. This over here is a chromatid. Chromatids are halves of doubled chromosomes. And we copy our DNA during interphase. In fact, more specifically, during the S part of interphase. S stands for synthesis. This is when we are synthesizing new DNA. So each of these doubled chromosomes has 2 sister chromatids that are identical and joined at the centromere. Don't confuse centromeres with centrosomes. It's easy to tell the difference. The centromere, which is in the middle, has an M here, centromere for middle. All right. So DNA will coil and condense and make the chromosomes visible when viewed through a light microscope. What that's saying is when we're actually using the DNA, when we're actually living the normal life of the cell and not in reproduction, the chromosome is all unwound and spread out. But we package them up real nice and pretty when we're doing cell division. So homologous pairs of chromosomes, one member of each pair is from the father, one from the mother. That means daddy gave you a copy of chromosome 1, gave you a copy of chromosome 2, gave you a copy of chromosome 3, etc. Mom gave you a copy of chromosome 1, chromosome 2, chromosome 3, etc. The only place where that gets weird is at the sex chromosomes. These sex chromosomes could have the potential to be different. But anyway, when we're looking at homologous pairs of chromosomes, they have the same genes, not the same version of the gene, that can vary, but they've got the same genes in the same locations, and that is the code for the same types of traits. Now, non-sister chromatids in homologous pairs, chromatids of one chromosome are non-sister chromatids of the other chromosome pair. What we're talking about is that we can, here we go. Non-sister chromatids exchange DNA with each other in a process called crossing over. Let's see if they have a picture of the crossing over. All right, they do not. So let me explain this real simply. When we are in normal cell division, I'm gonna see if we've got a picture here. If not, I'll go get one. Let's try that again. I was not liking the fact that the book is going through all this terminology and the pictures to go with it are mini slides ahead. What I did is I went and found a link to an Amoeba Sisters video. It has a side-by-side comparison, so you can see the difference in mitosis and meiosis. Anyway, so doubled chromosomes. Most people just say chromosomes, but if you had a chromosome and then during interphase you double the DNA, what you have is the original chromosome and the future, the new one. But as long as they're hooked together, we don't call them chromosomes. We call the original chromosome, when it's a part of a doubled chromosome, it is a chromatid, and this is the new chromatid. So chromatids are halves of a double chromosome. Chromatids are a future chromosome, and one of them will be the original chromosome, and one is the copy that is attached to it temporarily. So, sister chromatids. Each doubled chromosome has two sister chromatids. They're the same. They should have the same genes, same versions of genes. They're joined at the centromere. That's great. Homologous pairs are pairs of the same type of chromosome. So here's where we're going to talk about the difference in mitosis and meiosis. In mitosis, when you go through cell division, you line up at metaphase. Like prophase, you're going to, we're actually going to jump ahead to that picture. All right, so they're just doing they're just doing meiosis here, and that's fine because meiosis is what we're supposed to do. But with my hands here, I'll give you the difference. What you see is a pair of homologous chromosomes. Homologous is an important word. What they're saying is one is from daddy, one is from mommy, but they're the same kind of chromosome. Now, each one of those during interphase is going to double. So you have the doubled chromosome that you got originally from dad and the doubled chromosome you got originally from mom. Okay, great. Now, notice that at metaphase, they line up in pairs. You have the dad one and the mom one, the homologous pairs. It's not how you do it in mitosis. In mitosis, they line up single file, this one, and then the next would be underneath it, and the next one underneath it, next one underneath it. and then you would grow spindle fibers that attach to them. Actually, that happens before a tug of war. The spindle fibers is what moves everything to the middle. But in mitosis, you're going to rip the centromere, the thing in the middle here, apart. You pull until it tears, and you've separated the two chromatids. As soon as the two sister chromatids are separated, we call them chromosomes. So each one's a chromosome. That's mitosis. What you see here in meiosis, they are lined up in homologous pairs. That means that right here you have a double chromosome, here you have a double chromosome. You have 1, 2, 3, 4 chromatids. All right, 4 chromatids. The 2 chromatids of this double chromosome, the 2 chromatids of this double chromosome. When they are lined up that way, we call the 4 chromatids a tetrad. Tetra means 4. We call that a tetrad. We actually line up that way in prophase before we go to the middle in metaphase. So prophase of mitosis, you have double chromosomes floating around. Prophase of meiosis, you have homologous pairs of double chromosomes line up with each other. And then those two, this one and this one, they will be moved by spindle fibers to the middle at metaphase. So you see this in how we're lined up for the second metaphase, and then we tear that in half. That's what happens at mitosis. Okay, in meiosis in meiosis 2 we divide like mitosis. In meiosis 1 we do something special. Instead of tearing anything in half, we gently separate the pairs. Okay, so this pair and that pair, they're moved apart. But in meiosis 2, it's just like mitosis. Spindle fibers pull and we rip them apart. Okay. Let's go back. So here, homologous pairs are pairs that have the same genes, code for the same traits. They may not have the same version of the gene, same allele, but they've got the same gene. So gene for hair color will be a gene for a hair color on the other chromosome. It doesn't mean that they both say have red hair. Non-sister chromatids in non-homologous chromosomes. The chromatids of one chromosome, chromatids of the other pair. What we're talking about here is that this is when crossing over happens. I'm going to go to the picture. The Amoeba Sisters video I linked to for you has a really good video, but what we're talking about, let's see if there's a crossing over picture. There is not, but this will work really nice. So we have this tetrad here, these 4. You see the 2 in the middle, this chromatid and this chromatid, they are from different chromosomes, and they can swap pieces. That means where the book says, oh, well, this increases variability, genetic variation, da da da. Yeah, but they don't explain how. I'll tell you how. Because suppose these 2 chromatids that are right next to each other swap ends. That means that you would have the possible cells, because when we separate everything out, we're going to end up with I wish they had a picture at the end. Yeah, they do. We'll back up to here. Instead of having just the genes of dad, genes of mom, you could have a chromosome have the genes from dad, or the genes from mom, or these ones in the middle, they're going to swap ends. You could have most of the genes of dad, but some of the ones from mom, and most of the genes from mom, but some of the ones from dad, because they swap ends. That's a lot more variation. than just the two options of, oh, I got the dad genes I passed on, or oh, I got the mom genes I passed on. All right. Let's go back to crossing over. All right, so the non-sister chromatids can exchange DNA with each other in a process called crossing over. This happens whenever the doubled chromosomes are lined up in tetrads. Okay. Diploid means you got two copies of the DNA. In fact, I often say diploid because di for two. Okay, the di or bi means two. And you got one set of DNA you get from daddy and one set from mommy, so you have two sets in a normal cell. That is a diploid cell, a diploid cell. Okay, that has normal cells in your body have two sets. That means our goal in meiosis is to make gametes, cells that are haploid. They have half the normal amount of DNA. That means they have one set of chromosomes. So the sperm has one set, the egg has one set, and they come together, and you will have two sets, the normal amount. That's the whole point of doing this. So humans have 46 chromosomes normally. That's 23 pairs. And haploid cells, the eggs and the sperm, the gametes, they have 23 chromosomes. Okay. And when a sperm and an egg come together, you have two copies of that DNA. So meiosis has two divisions that we call meiosis 1. And meiosis has two divisions. We call them meiosis 1 and meiosis 2. I already told you meiosis 2 looks almost exactly like mitosis. Meiosis 1, you do things a little differently in the prophase, metaphase, anaphase. Prophase and metaphase, you form tetrads instead of being single file, instead of being floating around and then lining up single file. You float around in pairs and then you line up in pairs. And at anaphase in mitosis, you pull until you rip the double chromosome in half, rip the centromere apart. Meiosis 1 isn't like that. Meiosis 1, you have spindle fibers go to the double chromosome here, the double chromosome here, and they gently separate the two. Nobody gets ripped in half. All right, but you can watch the video if that makes more sense. So we're going through two divisions, but we only double the DNA at the beginning. That's why 46 normal chromosomes become 46 double chromosomes. I'm trying to show nucleus a little bigger. Become first cell division. You're down to two cells with 46 doubled chromosomes. Then you divide again to get 23. To get two cells here that are haploid, that are 23 chromosomes, two cells here that are haploid, 23 chromosomes. All right. So the goal is at the end to get to 4 haploid daughter cells. Oh, and we'll talk about this later, but the difference between spermatogenesis and oogenesis, the making of sperm, the making of eggs, when we're making sperm, we do equal cell division. So you end up with 4 sperm. Simple. When we're making eggs, we do unequal division. You still get the 4 haploid daughter cells, but only one of them is a functional egg. The others are kind of disposable, throwaway, just get rid of the DNA and get down to the correct number. Keep all the good stuff in the egg. And so the little throwaway cells, you have 3 of those, we call them polar bodies. You have 1 good egg. That's the difference in spermatogenesis, the making of the sperm, and oogenesis, the making of the eggs. All right. Both give you 4 haploid daughter cells, but oogenesis, only one of those is a nice functional egg. Anyway, each division you go through prophase, metaphase, anaphase, telophase. Straightforward. The difference is because we're going to divide twice, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Okay. And during interphase, in fact, during the S part of interphase, the synthesis part of interphase, all the chromosomes are doubled. Okay. During meiosis 1, homologous pairs are separated. In fact, I already told you that happens. They line up together at metaphase 1, and then they are gently separated at anaphase 1. So then you go from, I really don't like the way they're writing this, but it's fine. Okay. So you go from 2n, meaning you have double the number of chromosomes, down to n, the correct number of chromosomes, but they are doubled chromosomes. Okay. And that is not indicated in the way they wrote it. If you then during meiosis 2, the sister chromatids are separated, that would, let me say it a fancier, a simpler way, actually. During meiosis 2, you're going to line up just like mitosis. You're going to line up single file. There's no this pair and this pair, or this doubled and this doubled together as a pair. No, you line up single file. You grow the spindle fibers in. They tug of war. They rip the chromosomes in half, rip right through the centromere, separating this chromatid from that chromatid. As soon as you've got some light between them, as soon as you've torn them apart, we now call this a chromosome and this a chromosome. All right. So that's during meiosis 2. The sister chromatids of the double chromosome separate. Each daughter cell gets equal amounts of chromosome. That's the way it should be. It isn't always that way. We have abnormalities that happen, aneuploidies, but that's the way it should be. And so they're referring to these, the first division as a reduction division because you reduce the number of chromosomes, and the second one as an equational because you get equal amounts. It's a method. I'm not going to use either of those terms on the test, though. I don't like them. All right, so here we finally have the picture of meiosis. We have the normal cell. We go through interphase, the S part of interphase. We double each chromosome. We now have the X-shaped double chromosome, and each one of those has the original and a copy of it attached to a centromere in the middle. Okay. Then we're going to go through the first cell division, and we will gently separate the pairs. Okay. At anaphase, we separate the pairs. Then we're going to go through another cell division. We line them up single file, like there'd be a chromosome here, one here, one here. Right here, we're going to have 23 of those in a row in a human. Row down there. And then, just like mitosis, we grow the spindle fibers to the middle, we pull and we rip them apart. Okay, so the first cell division, we gently separate the pairs, the paired homologous chromosomes. The second cell division, we take that double chromosome and we rip it in half. All right. So in meiosis 1, the number of doubled chromosomes will be changed from 2n to n. It's still doubled chromosomes, though. Prophase 1 has events that you don't see in meiosis 1, don't see in meiosis 2. They're special. You're lining up in tetrads, groups of 4 chromatids, which is a fancy way of saying double chromosome, 2 tetrads, lines up with the homologous double chromosome, 2 tetrads. We now have 1, 2, 3, 4 double chromosomes. 1, 2, 3, 4 tetrads. Sorry. We now have 4 chromatids, which forms 1 tetrad. Okay. You can, during that time, doesn't always happen, have crossovers where the different tetrads exchange information, exchange DNA. That means that you don't just have the possibility of a chromatid that contains the genes you got from dad. You could have a chromosome that has the genes you got from dad, a chromosome that has the genes you got from dad, but some of the ones from mom on the end. A chromatid that has the genes you got from mom, but some of the ones on the very end are dad chromosomes. Or you could have ones from mom. If this is not clear, go watch the video that I linked to, the Amoeba Sisters video. Nice cartoon, good animation, explains it very well. This will result in unique chromosomes, mixtures of material. Yeah, good. All right, so meiosis 1, at metaphase 1, we are going to line up and separate line up in tetrads. And then at anaphase, we will separate the pairs. Okay. So during meiosis 1, tetrads line up randomly. That's totally true. The first tetrad here, you could have the dad chromosome, the double dad chromosome here, and the double mom chromosome here. The one underneath, it could be the opposite, where you have the doubled mom chromosome, then the double dad. And the one beneath that could be the same, or it could be different. So how this pair lines up has nothing to do with how the next pair lines up. We call that the law of independent assortment. And what I described is true for almost all species. Platypus are weird. We're not talking about platypus. Hold on just a second. Okay, so here's the weird thing. Platypus have 10 sex chromosomes, and all the male ones move together, all 5 males move together, all 5 female chromosomes move together. So they violate the law of independent assortment. There is a reason why, though, is that they all stack and stick together and then move like one super chromosome. So yeah, there are weird things in nature. But in humans, when you have a sperm, when you have an egg, it's haploid. That means it has 1 chromosome of each type. And that means for the sex chromosomes, you have either a sperm, you have either an X, I should say, or a Y. You don't have an X and a Y. There is a sperm with an X and there's a sperm with a Y. There's an egg with an X from this mom, from this grandma, and there is an egg with an X from that grandma. It's going to be an X because, well, we'll get into that in a minute. Not a minute, but a little bit. But the point is, if it's an egg, you should have an X and only one X. But it might be the X from this side of the family, this mother, or this grandmother, or from this grandmother. If you have a sperm, it could have an X or a Y. But it doesn't have both normally. There are exceptions. There are always weird things, always abnormalities in nature. But normally, a sperm has 1 sex chromosome. An egg has 1 sex chromosome. All right, moving on. Anyway, so metaphase 1, you line up in doubled and doubled as a tetrad. And then anaphase 1, you separate the two, separate the pair, separate the double chromosomes. Nothing is torn in half, you just separate the pair. All right. And it says the sister chromatids of 1 homologous chromosome, that'd be the 2 right here, are separated from the sister chromatids of the other homologous pair. That would be this one right here. So we separate the two. The two joined sister chromatids of each homologous pair go to the opposite end, and then we're going to squeeze in the middle and make them into separate cells. Okay, great. Now, at the end of meiosis 1, each daughter cell has doubled chromosome, 2 chromatids, this one and this one. Great. And they're still going to call it a chromosome. It would be much better if they would call it a doubled chromosome. I think that's just wise. Anyway, so this is showing the meiosis 1 division. You separate and you have 2 doubled, okay? Here you have the double for this chromosome, double for this chromosome. Now, if it was humans where we have 23 different pairs, you'd have 23 doubled chromosomes on this side and 23 double chromosomes on this side. All right, let's move on. Meiosis 2 is just like mitosis. This time we're going to line up the doubled in here, and then the next one here, and the next one here. We're going to line them up down the equator, down the middle during metaphase. And then we're going to grow the spindle fibers and rip them apart. Actually, we grow the spindle fibers and they do a gentle pulling, tug of war to line up in the middle, the middle, the middle. And then those spindle fibers get shorter and they pull harder and they pull harder until they tear the double chromosome in half. They separate the 2 chromatids by tearing the centromere apart. All right. So when we go through meiosis 2, that's how we make the haploid cells. That's how we make the cells that have half the normal amount of DNA. They're gametes. Those are future gametes. Those are going to grow up to be the sperm, grow up to be the eggs. All right. So mitosis gives us cells that have the normal amount of DNA for normal growth, normal repair, normal replacement of worn out cells. Meiosis gives us haploid cells, cells that have half the normal amount of chromosomes, and they're not all the same as what the parent had. They're unique combinations that neither parent had because you have some of your daddy's, some of your mommy's genes together. This crossing over gives a variability. The randomization, the independent assortment, that gives variability. All right, so here you end up with 4 not the same haploid cells that will become gametes, either sperm or eggs. All right, here we have an animation of interphase. I don't know why, probably just because they want to show the doubling of the chromosomes, which happens during the S part of interphase. Let's watch it. Are we going to do this? Great, they're moving towards the metaphase plate. All right, let's go to the next one. So we're going to separate the pair. Great. All right. So now let's do meiosis 2. Remember, meiosis 2 is almost exactly like mitosis. We're going to line up single file down the metaphase plate. We're going to tear the double chromosomes in half. Yeah. All right. I guess there's another video. I don't know why. Okay. Nice overview. I'm not sure if it's going to let us show this or not. I might have to remove it from the video. If so, just understand that there is a nice overview from the publisher, and you can watch it on the PowerPoints that you have access to. All right, so let's talk about anatomy of the male reproductive system. The male gonads are called testes. Obviously, the female ones are called ovaries. They are in the scrotum. The reason for the scrotum, the reason we have them below the core of the body hanging down, is because the core of the body is too warm for optimum sperm production. So we put them down there, and then we cool the blood that we're delivering from the body core with a exchange network called a pampiniform plexus that's in the spermatic cord. And this slightly cooled blood is what we use to supply them, give them oxygen so they can do the meiosis, make the gametes, make the sperm. So sperm is delivered to the body through a series of ducts. The epididymis, this is where sperm grow up. Sperm actually leave the testes as immature, unable to swim, and they pass through the epididymis from the head to the tail. It takes them about 20 days. And during that time, they mature, they become competent, know how to do things, know how to swim, I should say, not do things. And the tail of the epididymis is where we store the sperm that we are going to use in reproduction. The ductus deferens, that is a valid name. I will never use it in this class again. Every time I talk about it, it is the vas deferens. The reason why it's the vas deferens is because everybody's heard of a vasectomy. The ductus deferens, also called the vas deferens, during the vasectomy, you cut that. That's, if they cut it, if they seal it, then sperm can't move. Since it's a famous surgery, it's an easy way to remember the name of that tube that goes from the epididymis to the ejaculatory duct. So you have two of the vas deferens, one for each testis. They're going to go up, they're going to go over the bladder, they're going to fuse going into the prostate. And that's the ejaculatory duct that goes through the prostate gland. You'll see it in a picture in a second. And then you go through the urethra. The urethra goes from the bladder through the prostate gland. Think of the prostate gland as a donut below the bladder. So bladder is a chamber, holds urine, and it has a urethra that goes down through it, down from it, and that goes through a donut-shaped prostate gland. But that prostate gland has a hole through the backside, another hole. That's what the ejaculatory duct is going through. And inside the prostate, the ejaculatory duct and the urethra join together. And then the urethra, the same tube that carries urine out of the body, will carry the semen out of the body. All right, there are accessory glands, the seminal glands, the prostate glands, the bulbourethric glands. The bulbourethric gland, also called the Cowper's gland, this is what produces pre-ejaculate in common speech. We might call it pre-cum. The seminal glands and the prostate gland are what are part of the semen. We call that mixture of sperm plus the fluid from these glands the semen. All right, so there we have the picture. This little tiny gland here, that is the that is the Cowper's gland, the bulbourethric gland. That is where the pre-ejaculate comes from. And the purpose of that is a slightly basic, fairly liquidy mucus that lines the urethra. So it lines the urethra. What that means is from here out, if you had, say, a residue of slightly acidic mucus, you neutralize that. You make it so the acidity wouldn't mess with the sperm, because sperm are actually pretty fragile. They don't like pH change stuff like that. All right, so here we have the vas deferens coming up. It goes along the side and to the back of the urinary bladder. As it comes around the bladder, it is joined by fluid from the seminal vesicle. That's going to be future food for the sperm. I mean, actually, there are a lot of chemicals involved, but it has carbohydrates to feed the sperm as one of the things. So, as one of the components. So here, head of the epididymis, we have immature sperm. Tail, we have mature sperm. At ejaculation, we send sperm up the, give me back my mouse. We send sperm up the vas deferens, up through the vas deferens. When we get to the backside of the bladder, we join up with the seminal fluid, the seminal vesicle fluid, which is going to provide food. Then we go through this. This is the prostate gland. That's where I said the urethra comes through the middle and the ejaculatory duct comes through there. Okay. But the prostate gland is not just a area that there's a passageway through. This gland is a gland. It makes prostate fluid. There are lots of different components of prostate fluid, but the one I'm going to emphasize is that it acts like Red Bull for sperm. The sperm, even though they're mature sperm coming out of the tail of the epididymis, even though these are mature sperm coming all the way up, they don't swim until they get exposed to some of the components in the prostate fluid. That's why I say that it's like a Red Bull. It wakes them up so they can do their job. Anyway, moving on. All right, the scrotum and the testes, and of course we'll do the spermatic cord. We'll talk about things like the dartos and the cremaster muscle, shrinkage. Yeah, okay. So the scrotum is a sac of skin and fascia. It's hanging outside the abdominopelvic cavity. It's at the root of the penis, the base of the penis. It has the two testes. There's a septa between them. It is 3 degrees centigrade lower than the core body temperature. Why? Because we're trying to get the optimum temperature for making sperm. That's also why if someone has fertility problems, the doctor might say, hey, you need to change over to boxers. Your briefs or your boxer briefs, they're holding the testes up close to the body. They're too warm. You don't have optimum sperm production. If you want to have sperm, let them hang, let it hang, let the scrotum hang down, let the testes get a little cooler. Anyway, so yeah, there's a septum through the middle, and their lower temperature for sperm production. We've said all that multiple times. All right, the scrotum is affected by temperature change. This is where I said we would talk about shrinkage. It's not shrinkage on the penis, it's shrinkage on the scrotum, okay? When cold, the testes are pulled closer to the warm body wall. The dartos causes wrinkling, and the cremaster lifts the testes. So you have muscles around the scrotum that's hanging down. You have muscles that will pick up and lift them closer if it's too cold. You know, like if you're like, I'm in the polar bear club, I swim in ice water. Yeah, well, that's going to make your cremaster lift the testes closer to the body to get them warmer. The dartos, it wrinkles them? What? Well, if you think about it, if there's a smooth surface, say you're looking at the outside of the scrotum from above, and it's, I can obviously only do 2 wrinkles, one there, but this area that would be exposed to the cold water or cold air, a little bit of it has been removed. For every wrinkle, you have a little bit of the surface that you're holding away from the water. You might say, well, that's not much. You're right, but if you have enough wrinkles, it does have an effect. It does reduce some of the surface that is exposed to the cold. So there you go. Here we have the penis, okay? And well, I guess we'll talk about the penis later. And talk about the previews here later. But right now we have the testes in the scrotum, and each one has a spermatic cord. Hold on a second. Okay, so we weren't supposed to be talking about the penis. We were supposed to be talking about the spermatic cord and the scrotum. The spermatic cord is coming out the inguinal canal from the groin, because when the testes form, they form inside of the abdomen, and then they migrate downward. The testes, that's when they say, oh, they dropped. They're going down into the scrotum. Uh, they're inside of this spermatic cord. And let's talk about the cord. Let's talk about the scrotum, because that's their intent here. You have the skin of the scrotum, and below that you have a fascia that has the dartos muscle. That's the muscle you heard about that causes the wrinkling. When it's too cold, you do the wrinkling. Inside of that, and you can see the septum here, the septa that separates the two sides of the scrotum. Inside of that, we have the spermatic cord. The spermatic cord has fascia around it all the way around, and below that it has the other muscle, the cremaster that we talked about, that when it gets cold it will lift the testes closer to the body to try and keep them warmer. Below the muscle you have the vas deferens. You can see the head of the epididymis here. The tail is actually behind the testicle. But coming up from the tail, you have this cord, spermatic cord. Actually, spermatic cord would be in the middle there. Why are we not seeing it? Okay, they're drawing it right there, right next to the pampiniform plexus. This, I'm pretty sure, obviously it's in the spermatic cord, but I'm gonna check something. Hold on. Why did I do that? I don't need to stop the share. All right. Okay, so I was looking at the illustration in your PowerPoint and not particularly liking it because of where it puts the vas deferens. I was pretty sure that the vas deferens should be inside the pampiniform plexus, and it turns out that is indeed the case. If we're looking right here at the, this is the Encyclopedia Britannica, and it says, while ascending to the level of the bladder, the ductus deferens is surrounded by a network of arteries and veins, the pampiniform plexus, and nerves, and the whole thing is inside the spermatic cord. So that fits more what I was thinking. But so if you look at their own drawing of it, of course there's not. Do we have the whole thing or just that section? Yeah, they're not showing the whole thing. That's not helpful. Here we go. Their illustration shows it going behind, and that was freaking me out. And that's what your textbook was showing too. Let's go back to your textbook. Actually, let's go back here. So this is the type of drawing that's freaking me out, because the network of blood vessels is in front of the vas deferens, the ductus deferens. And I'm like, that's not what I'm used to. I'm used to being inside of. Well, the correct answer is probably, hey, it depends on what level you're at, because it certainly starts outside of the blood vessels, but probably ducks inside very, very early and then travels up inside the abdomen that way, inside of the pampiniform plexus. Anyway, so let's now go back to your let's go back to your book. All right, so I'm pretty sure it doesn't stay outside that far up. I would say maybe about there, I'm guessing. Anyway, so we have the inside the spermatic cord, we are going to have the tunica albuginea, albu like white, and then we'll have the tunica vaginalis. That is an extension of the peritoneum. Whenever the testes drop, they pull all this stuff with them. Inside of that, you have your epididymis, which is right next to the testes. That's going to receive the sperm in the head of the epididymis, and over a span of about 20 days, the sperm will grow up. By the time you get to the tail of the epididymis 20 days later, you're storing mature sperm there. Anyway, but this is special. This is magical. The pampiniform plexus, you have the testicular artery in the middle, and you have all these veins around the outside. What that means is that you have the artery going down through the middle, carrying blood to the testicle. And it's surrounded by veins that have already carrying blood that has already been to the testicle upward. Countercurrent exchange. What this means is that as the blood is traveling to the testicle, it's transferring some of the heat to the veins that are coming back from the testicle. That means it cools the blood off a little. It wouldn't make sense to drop the testes into a scrotum outside the body to keep them cool and deliver hot blood from the body core. So we use this network to cool the blood slightly before we deliver it to the testes, which is pretty amazing. Anyway, let's move on. So each testis is surrounded by two layers, two tunics. The tunica vaginalis is the outer one, and the tunica albuginea, it says, is the inner one. Each inside of each testicle, you have it's divided into little lobules. We could have up to 250 of them, and each one contains 1 to 4 seminiferous tubules. This is the site where sperm are produced. And if you're ever looking under a microscope and hey, I can't tell, what am I looking at? Is this a seminiferous tubule, or is this something else like I don't know, maybe the one of the collecting ducts in a kidney? How do you tell the difference? Well, the seminiferous tubules, the heads of the sperm are to the outside and the tails are pointing to the middle. If you see a bunch of those tails in the middle, clearly you're in a site of sperm production. You're clearly in a seminiferous tubule. All right, so the sperm go from the seminiferous tubules to the straight tubule. All the straight tubules come together to form the rete testis. A rete is a network, and the testis, because we're in the testicle. So a rete testis, you have all these straight things coming together. It's sort of like the fibers on a fishing net, you know. Now you remember efferent and afferent. Here we're using those terms again. We're going through the efferent ductules. In other words, sperm are exiting the testis and going into the epididymis. The epididymis, we have 3 parts. It looks kind of like a tadpole, and we call the top part where the sperm first arrived, the thickest part, the head, and then the middle is the body, and the narrow part at the end is the tail. A mature sperm stored in the tail until we're going to use them at ejaculation. It takes about 20 days to go from the head to the tail. All right, so blood supply. Testicular arteries arrive from the abdominal aorta. Testicular veins are from the pampiniform plexus surrounding each artery. This makes it cooler because it absorbs heat from the arteries. The artery dropping down to the testis is surrounded by a network of cooler vessels, cooler veins that are going up. Those veins that are cooler will absorb heat from the artery, cooling off the blood in the artery, and that means that cool blood is delivered to the testicle. So the spermatic cord has inside of it nerve fibers, the blood vessels we just described in the pampiniform plexus, and lymphatics that supply the testes. Here we have a cross-section drawing of a testicle and an epididymis, and you can see that the testicle, is divided into lobules with a septa between them. Each lobule has several, could be around up to 4 of these wiggly seminiferous tubules. They come together into a straight tubule, like the one you see here at the end of this lobule. And those straight tubules go into a network. Give me my mouse. Here we go. That we call the rete testis. This is the network. All right. And out of the rete testis, we then have the efferent ductule. That's the straight part before as we exit the testis and go into the epididymis. The head of the epididymis, the wide part here, the body would be the middle part here, and the tail is the end, the tail being where the mature sperm are stored. Okay, there's a section to show the same thing that we just saw in the pretty drawing. The pretty drawing is a heck of a lot easier to understand. Notice that the artery is in the middle, surrounded by a sheath of veins. That is the pampiniform plexus. Also inside of that, we have the nerve and lymphatics. And if you get far enough up towards the body, the vas deferens will go in through the side of the network and go up in the middle. All right, so let's talk about homeostatic imbalances, diseases. Testicular cancer is not common, it's rare, but the people who get it most often are ages 15 to 35. If you get mumps that are really bad, you could have inflammation of the testis. They call that orchiditis, and mumps can be a factor. Cryptorchidism means hidden, like crypto. Orchid, I don't know why orchid is the word they use for testis, but it is. But hidden testis means they didn't descend, they didn't drop, they didn't go down into the scrotum. If you have a painless solid mass in the testis, that's what they're talking about. 90% of the people have this problem. It can be cured by surgical removal of the testis that are up in the body, and often they follow up with radiation or chemo because cancer. Anyway, let's move on. So the accessory ducts, the ones we're about to talk about, the epididymis where sperm grow up, the ductus deferens, also called vas deferens, which is the tube that we carry the ejaculated sperm up towards the bladder, the ejaculatory duct that goes through the prostate, the urethra that also goes through the prostate. They join up in the prostate, and that passes the sperm to the urethra through the penis. Okay. So sperm grow up in the epididymis. The head has these efferent ductules that empty into the coil ducts of the epididymis. The body and the tail are the bulk of it, about 20 feet in length if they were stretched out. These are pseudostratified epithelium, lots of microvilli, stereocilia, which gives a surface area to absorb testicular fluid and to pass nutrients to the little sperm that they are nourishing and helping to grow up. All right. When the sperm enter, they are non-motile, meaning they don't know how to swim. They're not able to move themselves, and they pass slowly through the epididymis for about 20 days. At the tail end, they actually do know how to swim now. Not that they're going to do it without prostate fluid, but they can. And you could store, this will be an unpopular statement, but it is true. You could store sperm for several months before they would have a problem or die. During ejaculation, the epididymis will contract, and that will expel the sperm, that will squirt the sperm up the ductus deferens, up the vas deferens. So the vas deferens is about 45 centimeters long. It goes through the inguinal canal into the pelvic cavity. It expands on each side. The expanded area, and this is on the back of the bladder. The expanded area is called the ampulla. And at the ampulla, you have the vas deferens joining the seminal vesicle to form the ejaculatory duct. Smooth muscle in the walls of the vas deferens will move the sperm from the epididymis to the urethra. Think peristalsis type movement. A vasectomy is cutting and ligating the vas deferens. It is a nearly 100% effective form of birth control. If it does fail, it's usually because the people didn't follow the instructions. You are supposed to wait and wait a little while for healing, and then take a sample of sperm to the urologist. Sorry, a sample of semen to the urologist, and they check it to see if it has any sperm swimming in it. And not until the doctor gives you the all clear are you supposed to assume that you are now fully sterile. So, um, but some people are like, "Hooray! Let's have sex right now!" And then you still have some living sperm, or it wasn't closed properly, it didn't regrow, but you didn't take the time to do the test, so you don't know that there's a problem, and presto, there's the sperm. Okay, so we've already looked at this picture. Is there anything new? No, there's not. Okay. So the urethra carries both urine and semen at different times. Coming through the prostate, we have the prostatic urethra. The intermediate urethra is going up to the urogenital diaphragm, the where we leave the body, and the spongy urethra runs through the penis, and it opens at the tip of the penis at the external urethral orifice. That's an opening in the glands, which commonly people call the head of the penis. All right, moving on. So this is looking at the bladder from the back and above, kind of, because here we have epididymis, we have vas deferens carrying the sperm upward. You see they come upward, and what you're seeing right here, this is the ureter that carries urine to the bladder. Okay, and ducking underneath it, you have a now widened area in the vas deferens. That widened area is the ampulla, and the ampulla is going to join up with the seminal vesicle, and those together will join up into the ejaculatory duct that goes into the prostate and hooks up to the urethra. So yay. um so the three parts of the prostatic urethra, the intermediate urethra, and the spongy urethra. So give me my mouse. Prostatic urethra is in the prostate. Intermediate's this tiny little bit right here, and spongy is in this in the penis, in this spongy tissue. All right. So the penis itself, the male copulatory organ, our external genitalia are the scrotum and the penis, and the penis consists of a root and a shaft. The shaft ends in the glands. The glands is what people commonly call the head. The prepuce, or common name for it is the foreskin. That is loose skin that covers the glands. Um, in the United States, about 60% of US males are circumcised. That's where they cut away the foreskin. Um, Only 15% of males in other parts of the world. So this is very much a Western thing. But studies show that if you have the prepuce removed, there's a 60% reduction in the risk of HIV and a reduced risk for other types of STIs. All right, internally, we have two types of tissue. We actually have three columns of tissue, of spongy tissue. So the penis is made up of spongy urethra, named because it's going through spongy tissue, and three cylindrical bodies of erectile tissue of spongy tissue. This is it says spongy network, connective tissue, smooth muscle with vascular spaces. The one that goes around the urethra is the corpus spongiosum. That means the body of spongy tissue. And at the tip of the penis, that expands and forms the head, the glands. Um, and at the other end, it forks off and expands and forms the two bulbs of the penis. Those are internal to the body. Above that, you have a pair of tissues we call the corpus cavernosa. That means the cavernous, the big spongy tissue. The whole purpose of the spongy tissue is it fills up with blood. In us, meaning primates, the penis, we might talk in slang about, oh, there's a boner, but there's no bone in that. Now, there are in the animals that are in carnivora, but we're primates, and we don't have any bone in there. What we have is tissue that fills up with blood when the heart's pumping and you're excited, and then that tissue, you have around it strong layers of connective tissue. So you have the connective tissue tight, and then you have pushing against it, the blood rushing in, and that's going to fill up and actually pinch the vein that tries to draw the blood away. And so you get a traffic jam of blood, you get increased pressure, and it will make it stand up. That will make those columns stand up, which makes the penis stand up, become erect. All right, so an erection is when this erectile tissue, these three spongy bodies, fill with blood, causing the penis to get bigger, get more rigid. The crura, that's the body end, the proximal end of the corpus cavernosa, is surrounded by the ischiocavernosus muscle. This anchors the penis to the pubis, to the pubic arch. All right. Is there anything here we haven't discussed already? Oh yeah, here is the cutaway drawing showing the corpus spongiosum right there, and the urethra passing through the middle. And here we have the corpus cavernosum, the two cavernous bodies, the big bodies of spongy tissue. All right. The perineum is the diamond-shaped area between the pubic symphysis, the coccyx, and the ischial tuberosities. This is where we suspend the scrotum. It contains the root of the penis. It contains the anus. That's the area they're talking about. Right here, they have a drawing, and the perineum is, give me my mouse, this area right here. Okay. All right, let's move on. So these seminal glands, seminal vesicles, they're on the back side of the bladder. They have smooth muscle that'll contract and push their fluid out during ejaculation. This fluid is alkaline. That makes sense because sperm are wimpy and they die easily. That especially they could die easily in the vagina. The vagina is slightly acidic. In fact, the vagina has bacteria there that make acidity to keep yeast out. This is if someone gets a yeast infection, they lost their bacteria. Probably they had took a lot of antibiotics or something. Anyway, but that acidity, that would be enough. The acidity in the vagina would be enough to harm some of the sperm. So to protect those sperm, we one of the things we do is we have this alkaline seminal fluid, this basic seminal fluid. It's also full of fructose and citric acid and coagulating enzyme vesiculase and prostaglandins. The fructose, citric acid, those are going to feed the sperm. Those are going to help them have energy to swim on their long trip. Um, the the fluid from the seminal vesicles has a yellow pigment in it that will fluoresce under UV light. You've probably seen those things where they go into a hotel and they shine the black light, the UV light, to look, and oh, look, there's semen here and there. That's what they're looking for. This is about 70% of the volume in the semen. Seminal vesicle is the vast majority of the fluid, the volume of the semen. The duct of the seminal gland is going to join the ductus deferens in the ampulla and form the ejaculatory duct. All right, so that's fine. Then we're going to go into the prostate. The prostate will encircle the urethra just below the bladder. It's about the size of the seed inside a peach. It consists of smooth muscle that contracts during ejaculation. It releases a milky colored, slightly acid fluid. The acidity is like an unfortunate side effect. But what you're interested in, it contains citrate, contains enzymes. It contains a prostate-specific antigen. That's what people look for in the blood if they're looking for, does this person have prostate cancer? Look at for elevated levels of PSA. But it activates sperm, plays a role in sperm activation. That's why I said prostate fluid acts kind of like Red Bull for sperm. It will enter the prostatic urethra during ejaculation and 1/3 of the semen volume. Well, if you did your math, we had up to 70% of the volume from seminal vesicles, and then we had 33%, 1/3 from prostate. You might say, well, where's the sperm? What volume is that? It's a tiny, tiny amount. Obviously, these numbers are all variable a bit, but usually sperm make up like 5%, I think, of the volume of semen. Let's move on and see. Now, the bulbourethric gland, already said those are pea-sized, little tiny things below the prostate. Thick, clear mucus. I had that wrong. I said thin, but it's a thick mucus. It's clear. This is the pre-ejaculate or pre-cum. This lubricates the glands, the head of the penis, the tip of it, and it also neutralizes the acidity of the urine in the urethra. Because sperm are so wimpy about acid, they could die on the way out of the penis if we didn't do this. So we can neutralize the acidity. Um, Same picture, we got that. All right, homeostatic imbalance. So talking about benign prostate hyperplasia. Benign means it's not cancer. All right. This will affect 50% of men who are age 50, 80% of men who are age 70. Benign growth of the stroma and the glands of the prostate will squeeze the urethra, making it hard to urinate, leading to incomplete emptying. What this means is that a lot of old people have this problem. I've got to pee, I've got to pee, I can't pee, I got to pee, I can't pee, I got to pee, I can't pee. It's because the prostate in these men is getting larger and squeezing, constricting the urethra. Because they can't void properly, this could cause urinary tract infections or even kidney damage. So what kind of treatments do they have? You could take drugs to relax the smooth muscle and inhibit growth. You could have a surgical transurethral resection to widen the urethra. This used to be a really scary option because if you mess with the prostate, you might mess with the ejaculatory duct. A lot of men don't want you messing with their ability to ejaculate. So that was in the past problematic, but I'm hoping to read that things have changed. Uh, prostate inflammation of the prostate, prostatitis. This is one of the main reasons why people go to a urologist. And prostate cancer. This is the third most common type of cancer death in men. It affects 1 in 6 men in the United States. Digital exam screening, in other words, that joke about the doctor sticks his finger up the guy's butt to check their prostate, it's all true. That's the way it works. They palpate, they push to see if it seems enlarged. You can also look for levels of this prostate-specific antigen in the blood, but this has a lot of false positives. It's much more accurate to feel the prostate through the wall of the rectum and see if it feels enlarged. You can treat prostate cancer with surgery and sometimes it's radiation. It's a very slow form of cancer, so that's good. Um, there are metastatic prostate cancer, those if it has spread, you treat it with drugs that block testosterone synthesis or action. But yeah, this is a slow growing cancer. All right, so let's talk about semen. Um, milky white mixture of sperm and accessory glands. 2 to 5 milliliters of semen are ejaculated containing 20 to 150 million sperm. It contains fructose for ATP production. Remember, seminal vesicles have a lot of that, a lot of the fructose. That's food for your little swimmers. It protects and activates the sperm. Why? Well, because the 70% of it is this basic pH um, fluid from the from the seminal vesicles. Activate sperm. That's from the prostate gland waking them up. Fluids from the prostate gland. Red Bull for sperm. It wakes them up and facilitates sperm movement. This stuff is moving quickly anyway, and since the sperm are part of it, they get moved too. All right, and let's see. Alkaline fluid neutralizes acidity of male urethra and female vagina, enhances motility. All true. I already told you that the acidity in the urethra, the acidity in the vagina could harm or possibly even kill sperm because they're wimpy. All right, so characteristics of semen. It has prostaglandins in it that will decrease the viscosity of the mucus in the cervix. That makes it easier for the sperm to swim up through the cervical canal. Stimulate reverse peristalsis in the uterus. What I'm talking about is you have contractions in the uterus that will, you have the cervix dip trying to pick up sperm, and then you have the cervix contracting to push the sperm up inside, push the sperm up towards the fallopian tubes. Why? Because the fallopian tube is where the fertilization usually occurs. It's not in the uterus, it's further upstream than that, closer to the ovaries. Um, let's see, hormone relaxin and other enzymes that make sperm motility higher. You have ATP, again, an energy source to feed the little swimmers so they can swim, the little sperm. Uh, semen can suppress female immune response. Interesting. Antibiotic chemicals can destroy some of the bacteria, and clotting factors will coagulate the semen initially to prevent it from draining out, and then liquefy it through fibrinolysin so the sperm can finish the journey. What we're saying is that right after ejaculation, this semen is thick, and then over time, it becomes thinner. So the idea is that initially it's like, we made it, we're in the vagina, and it stays there. But then over time, it will get thinner and it'll be easier for the sperm to swim on. Okay. Part 2 is male reproductive physiology. Hold on a second. Okay, let's do male sexual response. This is around slide 70, 71, 72, 73, somewhere in there. All right. So erection is the penis getting larger and stiffer. The arterioles that carry blood to the penis are normally constricted, vasoconstricted. And that means they're carrying a little blood, but not much blood. Sexual excitement will make the central nervous system activate parasympathetic neurons. That will innervate the internal pudendal arteries, which will release nitric oxide. Nitric oxide will cause these vessels to vasodilate, and that will cause will cause relaxation of the local vascular smooth muscle. By the way, that's what Viagra does. It messes with the nitric oxide and causes dilation. So when smooth muscles relax, the arterioles dilate, the corpus cavernosum expands, it fills with blood, and when it does it kind of pinches off venous drainage. That causes engorgement, filling up massively of the erectile tissues with blood, and enlargement and stiffening of the penis because of it. The corpus spongiosum that the urethra runs through, when that fills up with this erectile tissue, fills with blood, it keeps the walls of the urethra open. This change is initiated by sexual stimuli. Could be touch, could be mechanical stimulation of the penis, erotic sight, sound, smells. It could be induced or inhibited by emotions or higher mental activity. So when we are propelling sperm through the male duct system, there's a spinal reflex from sympathetic nerves that serve genital organs. And this, now notice, this is sympathetic nerves. It was parasympathetic nerves that caused the vasodilation that caused the erection. But ejaculation is a sympathetic thing. Where was I? Here we go. Yeah, so sympathetic nerves serve the genital organs. This causes the vas deferens, the prostate, the seminal glands to contract and empty their contents into the prostatic urethra. The bladder's internal sphincter constricts because you want the semen going out of the body, not up into the bladder. This prevents urine from being released or prevents semen from going into the bladder, like we said. Okay, the semen in the urethra will trigger a spinal reflex through somatic neurons. The Cowper's gland, the bulbospongiosus gland, those muscles start to contract before the semen can get out. They cause, I take that back. Okay, they're saying and then the semen go out. But they should have said that before that the gland empties, and then we have our Cowper's gland, also called bulbospongiosus gland, um, producing that thick, clear mucus that lines the urethra. Anyway, an ejaculatory event is accompanied by, well, maybe that's why we're talking about it now, because this is ejaculation. That's pre-ejaculation. Anyway, the ejaculatory event is accompanied by intense pleasure that we call climax or orgasm. The heart rate goes up, the blood pressure goes up. After ejaculation, we have resolution. That's a period of time where there's relaxation. The arteries and the arterioles that are constricted by signals from the sympathetic nerves reduce the flow of blood into the penis at that point. Then you have a latent period or refractory period where you aren't able to have another orgasm. It could last a couple minutes. It could be several hours. The rule of thumb is it's shortest when you're youngest, and that period lasts the longest when you're older. Okay, so let's talk about ED, erectile dysfunction. Parasympathetic nerves of the penis may be not releasing enough nitric oxide, leading to difficulty in having an erection. 50% of men over age 40 have this problem. Temporary ED could be a psychological thing. It could be because you took too much alcohol, too much drugs, antihypertensives. That makes sense if we're trying to avoid hypertension. We're lowering the blood pressure, and you need blood pressure to raise the penis to make it erect. Antidepressants can do the same thing, cause erectile dysfunction. Chronic ED is entirely different. Chronic ED is a problem with blood vessels. You could have hardening of the vessels, arthrosclerosis. You could have varicose veins where the veins are distended and flabby. You could have a nervous system problem, a stroke, damage to the penile nerve, multiple sclerosis. Diabetes is often an underlying cause of erectile dysfunction. Diabetes can cause you to not be able to keep it up. New drugs like sildenafil, that's marketed as Viagra, it will enhance the effect of existing nitric oxide. I think what it's doing is blocking the removal of nitric oxide. Hold on a second. Okay, sorry about that, folks. So we're now going to talk about spermatogenesis. Genesis being the beginning, so this is the beginning of sperm. This is how we make sperm. The process of making eggs would be oogenesis. Spermatogenesis is how we make the male gametes. It happens in the seminiferous tubules. This begins at puberty, probably around age 14. And men make around 90 million sperm every day. That's good because sperm are kind of wimpy. Most of them are not going to make it. So tubules, we're talking about the seminiferous tubules. They have thick stratified epithelium, and they surround a central lumen that is full of fluid, and it contains 4 important types of cells. I want to point out, I've said this before, if you're wondering what you're looking at and under a microscope, if you see tails in the middle, that's a seminiferous tubule. So you could have cystanocytes. Those are cells, big columnar cells that sustain the developing sperm. They're support cells. Some people also call them sustentacular cells. It says, oh, they play a role in sperm formation. They provide the sperm with food. They feed them. They nourish them. They give them chemicals they need. Spermatogenic cells are cells that surround the cystanocytes and give rise to sperm. These are stem cells. These are primordial stem cells that are I shouldn't say primordial. These are stem cells that will create sperm. So you could have myoid cells. These are smooth muscle cells that contract to squeeze the fluid, move the sperm and the testicular fluid through the tubules. That obviously happens after we've made sperm. You could have interstitial endocrine cells, Leydig cells, and these make androgens, male sex hormones. They also make some estrogen. All right. So here we have sustentacular cells, sustanocytes. We have around them the spermatogenic cells, small little round nuclei. You can see the big columnar sustanocyte. The interstitial endocrine cells, those are located near these triangles, interstitial space. And actually, those would be the myoid cells, smooth flat cells surrounding the seminiferous tubule. Those are the ones that are going to squeeze. The interstitial cells, the ones that make endocrine, those would be this one, probably this one, maybe that one. Big nucleus, big nucleus. So here we have an electron micrograph showing the same thing. Remember I said if the lumen is full of tails, you are looking at a spermatic cord. Looking at a seminiferous tubule. And this is a seminiferous tubule. And you can see the lumen is full of tails of sperm. All right, so let's do a summary of events. This summary is 9 pages long, so it's not minor. Seminiferous tubules are in the testes. This is where we make sperm. Spermatogenic cells, remember, generate. They make sperm. They give rise to sperm. And there are 3 steps. The spermatogonia, those are the stem cells. They are going to divide to form 2 spermatocytes. And those 2 spermatocytes are going to make 4 thank you, dog. Those 2 spermatocytes will then they will modify, they will grow up a little, become secondary spermatocytes, which will then divide again to form spermatids. And those spermatids are what are going to grow up to be sperm. And I already told you, when we are making sperm, this means you're going to get 4 spermatids that are going to become 4 sperm. In oogenesis, the making of eggs, we still have 4 daughter cells, but 3 of those are going to be little polar bodies, and 1 of them will be the egg. Okay. So spermatogenesis begins at puberty. The spermatogonia, the stem cells that are touching the epithelial basal lamina, they divide through mitosis. And you might say to yourself, I thought this was all meiosis. Yeah, but we got to keep some stem cells around forever, so we make copies. At puberty, each division makes a type A daughter cell that will stay at the basal lamina, and it will maintain the pool. It will the stupid words, they're making it all fancy. You're going to divide in half through mitosis, and one of those is going to be the new spermatogonia, so you always have the ability to make sperm. The other one is going to make daughter cells, and those are going to move towards the lumen and divide to be primary spermatocytes, which will grow up to secondary and then divide again, blah, blah. Okay, so there it is. In meiosis 1, the primary spermatocyte will undergo meiosis 1, form 2 spermatocytes. Those will grow up to be secondary spermatocytes, which then you enter meiosis 2, they'll divide rapidly in meiosis 2 to become 2 spermatids for each of these secondary spermatocytes. So since the first division gave you two secondary spermatocytes, then you are going to end up with 4 spermatids. Spermatids are small, round cells, don't look like sperm, don't have a tail, midpiece. Round cells with large nuclei, they're close to the lumen of the tubule. Nearly all their genes are turned off, and DNA is compacted into dense little pellets. It's easy to transfer that way. So these 4 spermatids are going to grow up. They will eventually get longer, grow a midpiece, grow a head, or so grow an acrosomal process on the top of the what will be the head. These spermatids are haploid. They've got half the normal amount of DNA. In humans, that means they got 23 chromosomes instead of 46. That's the correct number we need for fertilization. These things still don't know how to swim. Then we'll go through spermiogenesis, where they take on a streamlined shape. They get longer, they get thinner, they lose a lot of their excess cytoplasm, they grow a tail. It's not saying it here, but they grow a midpiece full of mitochondria so they can swim. All right, so spermatids turning into sperm. You have the head, you have the midpiece, you have the tail. The head has a battle helmet on it called an acrosome, or an acrosomal process. This is full of hydrolytic enzymes. If this thing comes smashing up into an egg, it will try to digest its way in. That acrosomal process will burst open, spilling out the enzymes, and they will try to break down the protective the protective gel around the egg, so the sperm can get in. The mid piece is full of mitochondria that make ATP so you can move the tail. The tail is propulsion, it's how you move. All right, there we go. That's the making of a sperm. So, summary of events in the tubule. If things are going well, it'll take 64 to 72 days to make these spermatids. The sperm at that point are unable to swim, but you can the muscles pressing on. Let me back up. Right there, the myoid cells, the muscles, are going to be pushing on the fluid, moving the sperm and moving the fluid and the sperm it contains through the seminiferous tubule into the epididymis. And over 20 days, as they grow up in the epididymis, they will gain motility, they'll gain fertilizing power, the ability to cause pregnancy. So what do the cystanocytes or sustentacular cells, Sertoli cells, do? They go from the lamina to the lumen, and they surround the developing sperm. They form a tight junction, and that's important. We have a barrier to keep antibodies in the blood from getting to the sperm, because the sperm, we start making sperm later in life after we've already started making antibodies. We don't want to actually have the body decide those are foreign and weird and start destroying them. So the basal lamina, the basal compartment is tight on the basal lamina. This is where the spermatogonia, the primary spermatocytes, are. The luminal compartment is next to the lumen. This is also a tight junction thing. This is where the cells doing meiosis are and where the tubule lumen is located. All right, the tight junctions form a blood testis barrier. They prevent sperm antigens from escaping into the blood. That's what I was talking about, causing activation of the immune system. We've started making sperm later in life, after the immune system is all primed. These are weird. It's going to recognize them as weird and attack them if you let it. Oh, they're just saying it. Sperm is not formed until puberty, so is absent during immune system development. This results in sperm not being recognized as self. Therefore, we have to keep the sperm separated from the rest of the body to avoid being attacked by the immune system or attacked by the small dog with his unicorn toy who just jumped on my stomach. All right, moving on. So cystanocytes also provide nutrients and chemical signals to the dividing cells. They move the spermatocytes and spermatids from the periphery to the lumen. They secrete the fluid, the testicular fluid, that is full of androgens, full of metabolic acids, into the lumen that are used for transporting the sperm and giving them chemical signals. If you have cells that go haywire in this meiosis process, you eat them. The Sertoli cells will eat them and absorb the excess cytoplasm. Remember, we started with less cytoplasm, we got thinner, leaner. They'll produce two mediators to regulate spermatogenesis. Antigen binding protein will keep your testosterone levels high to stimulate production of sperm. Inhibin will inhibit spermatogenesis by inhibiting follicle stimulating hormone release by the anterior pituitary. So it will release one of the two of those depending on whether you want to ramp up or ramp down the production of sperm. Yes, you're a good dog, just too rowdy. All right, and here's the picture. I would definitely say look at one of those pictures, know it. I might label stuff, and here we have several similar pictures. Yeah, that's as far as I can go with that. So infertility affects 1 in 7 couples in the U.S. It's often caused by problems in sperm quality, quantity, or both. Over the last 50 years, we've had a gradual decline in male fertility. A lot of people think it's related to the chemicals we're exposing ourselves to, the food we're eating, or both. Some people believe the main culprit is foreign molecules we ingest, like environmental toxins, phthalates, oily solvents from plastics, or things like BPA, pesticides, herbicides. BPA is right here. The estrogen-like compounds that block action of male sex hormones. Yeah, they're right there. Antibiotics like tetracycline may suppress sperm formation. We might expose ourselves to radiation, lead, marijuana. Yes, marijuana can affect fertility. Or excess alcohol that could cause abnormal sperm to be produced. Two-headed or multi-tailed, they don't work well. You could have problems in calcium channels, hormone imbalances, oxidative stress, thermal related events, sperm over a lot of sperm. Sorry, thermal related events. Overuse of a hot tub could inhibit the ability to make sperm. That doesn't mean I would depend on hot tub use as a contraceptive, but it can reduce fertility. Production of gametes and sex hormones is regulated by hormones over time involving the hypothalamus, the anterior pituitary, the testes. This is what we talked about earlier, the hypothalamic pituitary gonadal axis. And this involves gonadotropin releasing hormone from the hypothalamus, follicle stimulating hormone and luteinizing hormone from the pituitary, testosterone and inhibin from the gonads. So here's the whole thing step by step. This picture, trying to see what it is. There we go. It's figure 2713. Yeah, 2713 has a 7-step. This is how the hormones work. I'll be happy if you remember which one comes from which location. On the next slide, we have a slide coming up where we'll talk about the change over a lifetime. So sequence of events. Hypothalamus releases gonadotropin releasing hormone. That binds to pituitary gonadotropic cells That makes them release follicle stimulating hormone and luteinizing hormones travel through the blood to the gonads. Follicle stimulating hormone stimulates spermatogenesis, so you make sperm. It does this indirectly by stimulating succintachocytes, the Sertoli cells. I'll stop fumbling over the syllables. It's the Sertoli cells, and what they do is release the antigen binding protein. Antigen binding protein keeps testosterone levels high near the spermatogenic cells, which promotes spermatogenesis. Basically, we're keeping the testosterone around, so we keep the testosterone levels high in the area, and they're like, ooh, well, we better respond to that. We better make some baby makers. We better make some gametes. Luteinizing hormone will bind to the interstitial endocrine cells, causing them to make the testosterone. Rising testosterone levels will stimulate trigger spermatogenesis. Testosterone that enters the blood will stimulate sex organ maturation, develop or maintenance of secondary sexual characteristics, and libido. Rising testosterone levels feedback on the hypothalamus and to say, stop that, we got it under control, we're doing the male thing. And the hypothalamus will inhibit gonadotropin releasing hormone, which stops the pituitary from sending the follicle stimulating hormone and luteinizing hormone. The cystanocytes can also release inhibin if the sperm count is really high. That's another way that we can inhibit gonadotropin releasing hormone in the hypothalamus, or a follicle stimulating hormone in the anterior pituitary. All right, so before birth, a baby has a testosterone level about 2/3 as high as an adult male. The very briefly early infancy, the blood levels drop and they stay really low through childhood. When you hit puberty, the levels come up again. And remember, the hypothalamus wants to release gonadotropin releasing hormone, have you make lots of sperm. You need higher levels of testosterone to suppress this until you have an adult pattern. All right, so we got that going on. Amount of testosterone and sperm produced by the testes, there's a balance from the hormones on the axis. It takes about 3 years to achieve during puberty, after which testosterone and sperm production are going to stay fairly stable throughout the rest of life. Without gonadotropin releasing hormone and gonadotropins, the testes will atrophy, they will shrivel up, and sperm and testosterone production will stop. So, okay, mechanism and effects of testosterone. Hold on. Okay, so if you don't have the gonadotropin releasing hormone, you don't have the gonadotropins from the pituitary, your testes are going to shrivel, your sperm and your testosterone production is going to stop. Wow, I should find that link. Yeah, I may throw a link up for you guys on the attempt to build a male hormonal based birth control. I heard some researchers being interviewed about this on the radio the other day, and it was really interesting because what they had to do was try to find a way that had low production of androgens in the low production of androgens in the testes themselves, but keeping serum levels of testosterone high. If you could have low testosterone in the testes, but high testosterone in the rest of the body, then you could have reduced fertility, but still have a normal, active, healthy feeling lifestyle. So, but they're working at it. Haven't pulled it off yet, but they're working at it. All right, so testosterone is actually a derivative of cholesterol. Some of the target cells will make cholesterol into testosterone. In the prostate, it's turned into dihydroxytestosterone and estradiol. Some neurons, that's what they do. Okay, brain neurons, they are chemical factors to do that. This dihydrotestosterone will promote spermatogenic. Sorry, where was I? The dihydrotestosterone in the prostate, the estradiol in the brain neurons promotes spermatogenesis, targets all the accessory organs. Lots of anabolic effects throughout the body. Let's talk again about the difference in anabolic and catabolic. Catabolic, the C should remind you of a pair of scissors cutting, breaking big molecules apart. Anabolic is the opposite. You're taking little molecule, and you're adding them together. So anabolic, we add things together, and we do it again and again. When you add things together, tends to require energy. So the catabolic, like your stomach breaking down nutrients, you get energy out of that. Anabolic, you tend to need energy because you're going to spend energy to get things done. So if you have a deficiency in testosterone, there'll be an atrophy in the accessory organs. Your semen volume will go down. You'll have trouble with ejaculating or erection. And the way we treat this usually is to give the person the way we treat the person is to give them testosterone. All right, so what are male secondary sexual traits? A secondary trait is one that you get at puberty, okay? This is features that we see in non-reproductive organs. They're mainly triggered by testosterone. One is the appearance of pubic hair, armpit hair, facial hair. Another would be growth of hair on the chest or other areas. The larynx will get bigger, causing the voice to get deeper. The skin will get thicker and more oily. The bones will grow. So bones grow, bones get more dense. Skeletal muscles get bigger, have more mass. This boosts the base metabolic rate. And that is the basis for sex drive or libido in males. Okay, and that is the end. So let's wrap this up and get it uploaded.